Are we live? Looks like. Yeah, let's continue. I think we went 10 questions in, 40 left. Yeah, we dived yesterday into immune system, and it's made from two systems, the slow and fast. Where is this? Chain of nucleotides. Nucleotides. Protein chain of amino acids. I want to find something regarding the immune system. Yeah, this one. Innate immunity and adaptive immunity. I think I got very good understanding of, I mean, good in quotes, right? But for adaptive immune system. But this one, I need to do something with my cough. It's so annoying. Cost and scale, yeah. Tell me more about vaccination only. I just want to double check that because great question. Have spotted something important. You are mostly right, but the answer has gotten more interesting in the last decade. Let me break it down. Oh, that's cool. Classic view. Vaccines target adaptive immune system. That was my understanding. Your immune system has two branches: innate speech.
Innate.
Okay. And adaptive is a fast generic first responder. Skin, mucus that's in the nose, right? Skin, I don't even know that. Stomach acid. Oh, stomach acid is a innate immunity. I need to find the hotkey for the mute button. Hold on. BS info mute. Control Shift M. No, it doesn't work. Control Command. Control Shift. No, it doesn't work. Hold on. Preference. Settings. Hotkeys. I see general. Hotkeys. Mute main. Unmute. Oh, nice. It's working. Hold on. The preference, hotkeys. Okay, until I get better, I will try to mute it really fast, so I'm not so annoyed with this. Okay, stomach acid is actually innate immunity that eat anything for region. Macrophage. Hold on. YouTube. Two years ago. This look like Venom, and then it's a release function.
He knows a lot about the science stuff. Professor Dave explains. For much of this series, we have been examining the innate immune system. Before we
That's perfect. Okay, so adaptive B cell, T cell, antibodies. Antibodies is protein that shapes to match antigens. That's what I'm recalling. Antigens and antigens, it's a proteins that's attached to the body of different pathogen means bacteria or viruses. And B cell produce antibodies. I don't know what T cell does. Let's see. Natural killer T cell. That's interesting. And this is innate system. Innate system, I don't even know innate system, like at all.
move on to the ever so captivating adaptive immune system, we have to get a closer look at some of the cell types we have been mentioning throughout the series. He knows a lot about the science stuff. Professor Dave explains. For much of this series, we have been examining the innate immune system. Before we move on to the ever so captivating adaptive immune system, we have to get a closer look at some of the cell types we have been mentioning throughout the series, each of which will get their own tutorial. First up, let's talk about macrophages.
Macrophages, long-lived pathogenic cell.
Macrophages are specialized, long-lived phagocytic cells of the innate
Phagocytes.
immune system. They play a pivotal role in recognition, phagocytosis, and degradation of cellular debris and pathogens. Macrophages also participate in tissue development as well as repair and resolution of inflamed tissues.
Tissue damage or infection. Tissue development, tissue surveillance, homeostasis.
Tissue development as well as repair and resolution of inflamed tissues. Additionally, these cells present antigens.
Macrophage. I'm trying to look for something familiar that I know and where I can like at least grasp some kind of base or something. But I see T cells, I have no function of the antigen presentation. So when Oh, this is pathogen, right? And sticking out is the antigen, and T cell is matching. Now the antibody should match.
To T cells, and they induce the expression of co-stimulatory molecules on other antigen-presenting cell types, which ultimately initiates the adaptive immune response. Furthermore, they are crucial in the initiation of inflammation by releasing pro-inflammatory cytokines and chemokines, which in turn recruit other immune cells to inflammation sites. Macrophages were first discovered late in the 19th century by Ilya Mechnikov, and they are evolutionarily conserved phagocytes found.
Thank you, Ilya. Ilya Mechnikov. Respect goes to you.
By Ilya Mechnikov, and they are evolutionarily conserved phagocytes found in vertebrates. Initially, scientists proposed that all tissue macrophages originate from the circulating blood monocytes.
So this part. Imagine if like one day I fully understand this. And not just understand, can actually explain it. In a reference to like basic world, like everyday people know what's up. That would be so cool. I can't wait.
Which continuously migrate to different tissues and differentiate into macrophages. However, recent studies have overturned this prevailing paradigm, as several independent groups reported the results of genetic lineage tracing studies in mice. They provided strong evidence that resident tissue macrophages are in fact established during embryonic development and persist into adulthood independently of blood monocyte input in the steady state. Despite the dramatic progress made in the field of macrophages,
I feel like this video is great. By the way, thank you. Subscribe if you don't. If you didn't. But I think it's way over my head right now. Like it's so many, like I don't understand what he's saying. Let me try this one.
Hi, this is Tom from ZeroToFinals.com. This video was designed to give an overview of immunology, so that you understand all the basic features of the immune system. It will also help you to return to this video once you've already learnt the details, so that you can again fit those details back into the bigger picture about how the immune system and the that are already at the location of the infection and respond immediately with a general system can be categorized into the innate immune system and the specific immune system.
Why he's saying specific? I thought it's adaptive.
The innate immune system consists of the components that are already at the location of the infection and respond immediately with a generalized response. The specific immune system involves specialized T and B cells that are waiting in places away from the site of infection, and then when they're activated, they launch a specialized...
So they actually located in the lymph, right? In this place, where is it? Yeah, I wouldn't find it, but oh, we still have research for tomorrow, yeah.
System that fights the specific invading pathogen. The specific immune system takes a little while longer to recognize the complement system that works alongside the innate and specific immune systems. Let's look at what happens when a pathogen invades the body. Initially, there are physical barriers.
So pathogen is mean like something external, something that's not belong to the body, right?
That need to be broken before an infection can take place. The skin is an example of one of these barriers. Most invaders will get stuck at this barrier. However, occasionally an invading pathogen will get through, such as when there's a cut in the skin.
Hello?
As well as the skin, there's other physical barriers, such as the mucosa of the respiratory system, the gastrointestinal tract, and the urinary tract. There are also chemical barriers that help destroy harmful pathogens before they can cause any infection, such as the hydrochloric acid in the stomach, lysozyme in sweat and tears, and lactic acid in the vagina. Let's look at the initial responses of the immune system once an invading pathogen has broken through the physical and chemical barriers to infection. 3 things happen. Number 1, macrophages recognize the pathogen and activate the innate immune system.
How? That's my question. How does it recognize it? Because I thought the adaptive system recognized it by antigens that's sticking out, and then antibody get deployed. But how macrophage recognize the pathogen? This was dogma until the 2010s. Whoa, recent. When researchers started noticing something with kids who got BCG vaccine, tuberculosis vaccine, had lower overall childhood mortality, and not just from TB. They were also more resistant to respiratory infection, unrelated viruses, and even some cancers. That's interesting. This led to the discovery of trained immunity, innate immune cell, especially monocytes, macrophages, yes, and NK cells can actually be trained to respond more aggressively to future threats, even unrelated ones. The mechanism is different from adaptive memory. It's not antibodies or T cell receptors. It's epigenetics, rewriting of the cell DNA packaging. Whoa! This is so cool. So innate immunity react faster than the trained one, right? I forget the second name again. Innate and adaptive. So, and just recently we found out that actually innate immune system can affect epigenetics that will actually alter DNA to react more aggressively on the invaders. This is so cool. Make certain inflammatory genes easier to switch on next time. This training lasts weeks to months, sometimes up to a year, much shorter than adaptive memory, which can last a decade. Like this session stuck on the vaccines, but
Number 2, dendritic cells pick up antigens from the pathogen and then head off in the blood and lymphatic systems to track down the relevant T and B cells of the specific immune system and activate them. And number 3, the invading pathogen activates the complement system directly via the lectin pathway and the alternative pathway.
So dendritic cells, I'm assuming that's our brain, right? And it's picked up antigens, meaning this stuff, and going into our lymph system, track down T and B cells and activate specific system. Remember how I was saying the economics water machine? I was off, way off. But what happened, actually brain track it. And again, pick up antigens, like how? I have so many how questions, but
pathway, and we'll talk a bit more on those later. First, let's look at how the innate immune system provides an immediate response to infection. The first to respond are the macrophages. They recognize pathogens by specific characteristics that occur on pathogens but don't occur on cells of the body.
Okay, I'm so now, so like everyone recognizes it by antigens, even macrophages. But
These characteristics are called pathogen-associated molecular patterns, or PAMPs.
What? Just yesterday I learned that that's called antigens. 100%. Somewhere here. I remember. Okay, let's switch.
They recognize PAMPs using various receptor types, including toll-like receptors. At this point, I need to talk briefly about a process called phagocytosis. Now, phagocytosis is the process that macrophages and other phagocytes like neutrophils use in order to destroy pathogens. So what happens is, once they've recognized the pathogen, they start to wrap their cell membrane around that pathogen and absorb them to within inside their cell. The pathogen is then left inside something called a phagosome, and this phagosome fuses with surrounding lysosomes that provide digestive enzymes that then break down that pathogen, destroying it, and also processing all of the components into harmless waste products. If the invading pathogens are harmless enough,
So it's basically engulfing it. And what's the name of the part?
destroy the cell, then what is called phagocytosis.
Phagocytosis. Phagocytosis. Phagocytosis. Phagocytosis.
Now, phagocytosis is the process that macrophages and other phagocytes
Macrophages. Oh, phagocytosis I added yesterday? Wow. Cell eating, yeah. Macrophages. Macrophage.
like neutrophils use in order to destroy pathogens. Outside them alone, then they're cleared and it goes no further. Harmless waste products. If the invading pathogens are harmless enough that the macrophages can deal with them alone, then they're cleared and it goes no further. If the attacking army is too great, the macrophages need help, so they release cytokines, which are signaling proteins, sort of like local hormones, that sound the alarm of an infection in the local area. This leads to a process called inflammation. One of the most important actions of these cytokines is to recruit and activate more cells of the immune system, such as macrophages, monocytes, and neutrophils. Monocytes are precursors to macrophages that float around in the blood. Once they enter tissues, they differentiate into macrophages and can then carry out all the processes that macrophages normally can.
Okay, so monocycle, they floating in the blood. Macrophages, they actually in a tissue. This one precondition. Inflammation, it's a signal by releasing macrophages release cytokines, which is inflammation, to get a signal to recruit more of themselves. But they all not in the blood. I mean, this one in the blood, this one not. That's my understanding so far.
Carry out. Neutrophils are another type of phagocyte that circulate in the blood and can enter the tissues and help by destroying invading pathogens. The inflammatory response also involves a number of other processes that help to contain and fight the infection. These are vasodilation, increased vascular permeability, mast cell activation and degranulation, releasing more cytokines that further stimulate the inflammatory response, activation of the clotting system, and activation of the kinin system. Inflammation itself actually stimulates macrophages and neutrophils to secrete more cytokines, notably chemicals called interleukins. This is known as the acute phase response, and it leads to a more systemic inflammatory response. This involves sending cytokines, specifically interleukin-1, to the brain to tell it to produce a fever that leads to high temperatures that are
interleukin go to the brain to produce fever.
Poorly tolerated by many pathogens, and cause interleukin-1 to the brain to tell it to produce a fever that leads to high temperatures that are poorly tolerated by many pathogens, and
okay, so the high temperature actually killing the pathogens? That 2-degree difference from our normal temperature to like 39 Celsius or like 105 actually enough to kill pathogens? Like, why don't we just go to sauna and cook ourselves for a little bit? I want to ask that question. If immune system send a signal to the brain to innate immune system send a signal to the brain to increase temperature because many pathogens cannot tolerate couple degrees higher than our normal body temperature, human normal body temperature. What is it? Same effect can be reached just to go to sauna and sit in a very hot environment for like 20 minutes. Yeah, that's called fever. Hello, yeah, I know. Pyrexia medically. The mechanism works like this. When immune cells detect pathogens, they release cytokines IL-1, IL-6, TNF alpha, which signal the hypothalamus to raise the body thermal set point. Yes, I was aware of this from the movie about cells. Really good movie, like old one, but very good one. Your body then actively generates heat through shivering and vasoconstriction until it reaches a new target. Many bacteria and viruses replicate poorly at elevated temperature. Oh, they replicate poorly. And immune cells actively, specifically T cell proliferation and neutrophil function increases with heat. Now for the sauna question. This is a common misconception, and the answer is no, not really the same thing. A sauna causes hyperthermia, which is mechanically different from fever. Fever, you rise to the set point and your body actively works to reach it. The whole system is coordinated around the higher temperature. Sauna hypothermia, external heat, forces your body's temperature up while your hypothalamus is actively fighting to cool you down. Sweat, vasodilat, your set point state. Yeah, I understand that set point state 37. I was asking if the pathogens would die, but they actually not die from the high temperatures. They decrease, they slow down in reproduction, right? The key difference is that during fever, the elevated temperature is accompanied by a coordinated immune response. The cytokine cascades that cause the fever is also orchestrating increased immune cell activity through your body. Sauna heat doesn't trigger that cascade. Okay, so the fever, it's not like fever, the temperature itself killing the pathogen. It just signaling mechanism for the whole body to generate more. What's the name of the cell that's engulf everything? We learned it today, right? Macrophages. To create more macrophages from innate immune system. The key difference second is that fever also accelerates in increased immune cell activity through your body. Sauna heat doesn't trigger that. That said, sauna use does have a real health benefit. Yeah, I'm not talking about the health benefits. I understand that sauna is good. I was trying to figure out what actually kill the pathogens in our body. We have a fever as a response, and from your explanation, how I understand the fever is actually more of a signal mechanism for the whole organism to go into fight mode, create more macrophages, so it can engulf pathogens. So temperature is more, or fever is more a signal for the body, not actually killing a pathogen from burning them, right? Or by breaking them up. It doesn't break the pathogen from the temperature. Therefore, just by rising the body temperature, yes, I know that internal thermostat set to cool it down. That's logical. But just by rising the temperature, it will not kill a pathogen that's inside of your body. And go to sauna will not trigger immune system, will not create more, I keep forgetting that word, macrophages. Will not create more macrophages, therefore you're still sick. Is there somewhere close? I'm trying to be good at muting myself and I'm coughing. Why I'm asking that? Because my wife yesterday asked me, like, is the fever actually the temperature itself kills the pathogen? And I thought that's no. But when she keep drilling me, I thought like maybe yes. So that's why I'm investigating it today. Correct. Let me sharpen it a bit because there are small nuances. What fever actually does to pathogens, the breakdown. What fever actually does to pathogen, the break. So it is breakdown of pathogen. Direct thermal killing, mostly no. Oh, okay, so direct thermal killing, mostly no. You're right, the couple of degrees above 37 doesn't cook or structurally break. Yes, exactly, yeah. Now it's clear. Okay, kids, listen. A couple of degrees above 37 doesn't cook or structurally break most pathogens. Period. Human fever tops out around 40-41 Celsius. Most human pathogens evolved specifically to live in us, so they tolerate our normal range fine. Blah, 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 blah. To actually denature bacterial proteins or destroy viral capsids thermally, you need temperature well above what living human body can reach, thinking 60 or Celsius for pasteurization. Okay, I was right. Growth inhibition, particularly, yes. This is a small nuance. Some pathogens do replicate more slowly at 37. Yeah, so basically, fever, it slowed down the replication. Rhinoviruses are classic examples. They prefer cool temperature. Immune signals, and this is the main event, you have got it right. The actual killing done by your immune cells, macrophages, engulfing pathogens. Neutrophils release antibiotical compounds, cytoxins, T cells. Okay. Now the question, macrophages. When I'm getting a fever, I know it will be gone as soon as all pathogens are gone, right? So I need more macrophages. How can I accelerate production of macrophages? How can I chicken soup? Good question. The hardest part is mostly you get it out of your immune system way and let it do its job. Oh, get out of your immune system way and let it do. There is no magic lever to crank a macrophages production, but the real factor that is a support or undermine the response your body is already mounting. What actually helps? Sleep, hydration. Don't suppress the fever unnecessary. Protein intake, zinc and vitamin D. Moderate movement if you can tolerate it. What actively cures? Alcohol, poor sleep, high stress, cortisol, smoking, overexertion. I didn't know that. Like, now I see clearly, and now I will remember for my life, like, what's good, what's bad, and I know why. Because we just need more. I keep forgetting that word. Macrophages, macrophages.
Cause reduced appetite and lethargy so that the person conserves more energy that can be used to fight the infection. It also involves sending interleukin-6 to the liver to produce acute phase protein that act as something called opsonins. And remember this word because we'll talk about opsonins in a minute. Additionally, interleukin-8 is released that recruits and activates more neutrophils. Interleukin-2 and 12 activate natural killer cells, and tumor necrosis factor alpha is released that does all of these effects by itself. As I just mentioned, I want to take a quick look at opsonins, as these are very important in the immune system. Opsonins are complex molecules that attach themselves to pathogens and make it easier for macrophages and neutrophils to recognize and phagocytose that pathogen. I think of them a bit like a fork that spears the pathogen so that macrophage can attach to the other end of the fork and then use it to eat the pathogen. An example of an opsonin that you may have heard of is C-reactive protein.
That's pretty cool. Opsonin, it's a handlers that help to hold pathogens for macrophages to eat them. Macrophages, I remember them now. Macrophages is the topic of this stream today. Rename it to macrophages.
Or CRP. CRP is produced by the liver in response to interleukin-6, and we actually measure the level of CRP in our patients to assess how much inflammation there is in the body, and it can be a really good indicator of the severity of the infection that our patient is suffering with. So that summarizes the innate immune system, a generalized system that recognizes and responds to an invading pathogen by causing an inflammatory response and recruiting cells that destroy the pathogens by phagocytosis.
Okay, thank you for summary. And I still don't know how the T cell works.
Research, the dichotomy between embryonically derived and adult-derived macrophage populations does not fully encapsulate the complexity and diversity found among tissue macrophages. Depending on the tissue microenvironment, macrophages exhibit plasticity in terms of phenotypes and function. Macrophages can infiltrate various tissues under inflammatory or non-inflammatory conditions. They can assume different functions and phenotypes according to the cues they receive.
By the way, I don't know why, but now it's easier to understand this video. Now I know what he's talking about. What's macrophages as a cell that eats the other pathogen by engulfing it. It can go to different tissue under two different conditions, inflammatory or not. That's when we have a special signals in a blood. I get maybe not blood, or maybe blood, right? So and signals are IL-4, IL-13, so on.
Receive from the microenvironment. Broadly, macrophages can be divided into two distinct subtypes. One, classically activated or M1 macrophages, and two, alternatively activated or M2 macrophages. Let us discuss how these two subtypes of macrophages are formed. In vitro,
this is pretty cool because like macrophages could be a very good way to remove something from the body. And in the immune system, it's pathogens, right? But imagine if your blood vessels are like full of cholesterol and you need to remove it. You just not train macrophages, but you mutate macrophages into function to actually eat that cholesterol, the bad cholesterol, and remove it from the body. Chpak, and you clean. You're 14 years old again. Or even better, we need to train macrophages to do what blood transfusion does. When you take a blood of young people and putting it in the old body, and the old body regenerates, basically it become younger.
M1 macrophage.
I actually gonna ask this. DNA, so should I continue here or not? Yeah, I will continue in this question. For the two researches, I'm gonna create a new chat and go with the project biology science. Okay, looks like macrophages are attacking the pathogens and remove them or destroy them into different parts by engulfing them and then destroy them, right? And turns out that recently, or maybe not recently, there is different types of macrophages, M1, M2, and so on. So do deep research and do a reality check and let me know what you think about idea that we take macrophages, we reprogram them, create completely new macrophages that's actually attracted not to the pathogens, but to the special things that we're gonna call it specimen A. So macrophages will attract to the specimen A. And what is a specimen A? I want to connect this idea to the study, not sure how recent that, but when you take a blood of the young mice and putting it in the old mice become younger significantly, and it's proven. And even it was a startups like this when it's very expensive, but you can get I don't know if it's called blood transfusion or not, but you can basically get a young blood as the IV, and you feel much better, and your body perform much better. And the scientists try to figure out what actually in a young blood that helps such a boost in health and performance of the old body. And I believe that they ended up to conclusions that it's not what exists in a young blood, but what doesn't exist in a young blood. So basically, in the old blood, certain things accumulate with age, like toxin, like it's not toxins, like something they didn't know. And we call it specimen A, accumulate in the old blood. And if you remove it and your blood become young again, then you become, the whole body become young. So the idea is we don't need to do those blood transfusion anymore. We can just train macrophages to remove that bad stuff or remove pathogen, specimen A, to remove not pathogens, but we can reprogram macrophages to remove specimen A from the old organism blood to become young again. Let's have it do it. Done and done. Good luck. Fascinating idea. It sits at the intersection of macrophages biology and parabiosis young blood research. Parabiosis. Oh, Conbay, Wayscorer, Rubin Labs. Yes. Bro, excited. And the emerging, I don't know this one, but blood factors dilution field. Thank you. Yeah, give me more of those complicated words. There's a real science here worth pulling on. Yes, let's do it, especially around GDF. Yeah, of course, guys. How can we forget about GDF11? B2 microglobulin. I don't know, vocabulary 2020 convoy. Neutral blood exchange. Study that arguably supports your delusion, not addition hypothesis. I guess that was my hypothesis. Before I dive in a couple of clarifications of the research match what you actually want. How technical should be the report? How technical the report should be? Depth report and scope of specimen A. Focus on the feasibility angle. Do you want me to include the state of engineer CAR macrophages therapy? Oh, they actually engineer macrophages? I was just guessing that you can engineer macrophages and teach it to do different stuff and assess whether it could realistically be a startup thesis or keep it purely to biology. If you if you'd rather I just go, say, just go. I would say 3.
Next, let's look briefly at the complement system.
Sorry.
Macrophages are polarized by Th1 cytokines, such as colony stimulating factor, tumor necrosis factor alpha, and interferon gamma, either alone or together with lipopolysaccharide from bacteria. M1 macrophages express pro-inflammatory.
Professor Dave, you are amazing. I don't even know how you comprehend all this.
Cytokines such as interleukin 1 beta or IL-1 beta, IL-6, IL-12, IL-2 are polarized by Th2 cytokines such as IL-4 and IL-13 and produce anti-inflammatory cytokines such as IL-10 and transforming growth factor beta or TGF beta. It is important to note that macrophages can adapt special tissue microenvironments to polarize specific phenotypes such as tumor-associated macrophages or TAMs and adipose tissue macrophages or ATMs. Each phenotype of macrophages has a relatively specific expression of.
Okay, I have another idea. And okay, calm down, guys. I know. All my ideas is basically steps that's already been taken by scientists and already exist in life. But again, I'm catching up, right? So with those 42 researchers, if you miss the previous sprint, we're going against these 242 researchers that we need to meet, confirm. So the idea is in biology, let's start another one. New chat. Research. I actually need to create a pipeline. So every time when I have an idea, it just goes through the pipeline, validate it, see if it already exists, blah, 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 and it goes through all stages. We're gonna draw that pipeline. We're gonna draw that pipeline tomorrow and create it the day after, right? At the project. So in biology, obviously there's a lot of different signaling, right? The way how our body regulates everything between cells, like take immune system, it's crazy. It's release different signals to signal of production of macrophages, and there is 2 different types. And they release in different like IL-10, TGF beta, IL-1 beta. And we trying to accurately map all those different signals and what they cause in it. And there are so many different variations of those signals. And we try to precisely map what each signal would mean or cause. But what if there is no precision there? What is like, maybe it's like language. We try to communicate intent of cell or immune system try to communicate intent as, hey, I need to do this. And it's basically express itself with different sentences like of urgency, like, hey, do it right now. I really need it badly, more of this, right? Like you can use different words and sentences to describe this you need more. And maybe all this IL-4, IL-13, and so on It's just different words that's slightly different, obviously, since the intent a little bit different, urgency, but mostly intent, then reaction of the other parts, the reaction of the other mechanism involved in that communication, different. But for scientists right now try to map it like one protein, like this protein causes this protein causes this, or like combination of these two proteins. What if we just try to map intent, not individual worlds? Bonds? No, words. Oh, my English is bad.
Beta. Each phenolize specific phenotypes, such as tumor-associated macrophages, or TAMs, and adipose tissue macrophages, or ATMs. Each phenotype of macrophages has a relatively specific
That just reminds me how to get money, ATMs.
expression of some cytokines, chemokines, toll-like receptors, or TLRs, and matrix metalloproteinases, or MMPs.
This is so over my head, like literally, like it's like Japan, like someone talking Japan to me.
The M1, M2.
Sorry, not Japan, Japanese. Like I didn't even finish learning English, and I'm trying to learn biology in English.
Paradigm is unfortunately not sufficient to address all polarized phenotypes that macrophages can assume. This is because, depending on the
Phenotypes, so those are signals, okay?
stimuli, they have the ability to modify their polarization status.
Polarization status, that's what they might define, okay? And that polarization means from physics when like literally light polarize.
For example, macrophage phenotype can change during tissue repair, switching from pro-inflammatory phenotype, or M1-like, to an anti-inflammatory phenotype, or
This is cool. I actually never knew how tissue repair. So macrophages, it's also repairing the tissue. Can we like be what's the name of X-Men? Wolverine? What a rich idea. You're essentially asking whether cytokines signaling is better understood as a combinationary language or intent rather than lookup table of molecular X effect Y. Yes! Finally someone understands me. Like I just rumble, you heard me, right? And it's actually created a tweet for me. And now my idea, sorry, not my idea. I'm just brainstorming here, right? I'm bounce idea against the wall. But now it can be posted on Twitter and someone understand me better. Like this is the power of AI. I love it so much. Like it would take me like 10 minutes to try to explain what I mean, and I just take it and doing it in one sentence. This actually connects to some real emergent thinking in immunology. Cytokine, pleiotrope, redundancy, context dependency, and to information theoretics approaches to cell signaling. Yes. Yes. Before I dive into deep research, a couple of quick questions to make sure I pull the right threads. Which angle matter most? I think philosophical, right? The methodology, how would we actually map intent instead of individual signals? Information theory, ML on... Oh, I like this too. Combination system biology. Scope on signaling system. Focus just on the immune system or include other cell communication too. Yes. Hormones. Yes. Just focus on immune system. Depth of existing work. Do you want me to dig into specific researchers lab already pursuing combination versus signals? Yes. Let's go.
M2-like. Also, different polarization and activation markers of M1 and M2-like macrophages can coexist in tissues at the same time. Therefore, it is extremely important to be aware of...
Okay, bathroom break.
of the cellular and functional plasticity while studying macrophages in vivo, such as in diseases. M1 macrophages express high levels of major histocompatibility complex class II, or MHC2, CD68, as well as CD80 and CD86 costimulatory molecules. M2 macrophages...
I'm wondering why everything's supposed to be so complicated. like why we try to name every single molecule. I mean, it's probably important, but then when I'm listening, it just like stream of noise to me because I don't understand it. And yes, we create labels to compact bigger concept into one word, but the way how we name every single molecular structure, it's insane. I want to say, like, do we need it? I want to question, like, do we need to name every single molecule? Maybe, right? That's why they did the protein folding, and every single one, like, have different properties.
Express low levels of MHC2 while featuring expression of CD163 and CD200R membrane glycoprotein, as well as high levels of MGL.
I'm so lost. Like, we don't have to, like, there's no need for watch this one any further without knowledge because I have no idea what they're talking about.
1 and family. Both phils, acidic. Again, macrophages are phagocytic in nature. Other notable phagocytic cells are granulocytes, consisting of
Phagocytic means the one who is engulfed, right?
Surround
Phagocytosis. Yes, I learned two things today: macrophages and phagocytosis. So what he's saying, macrophages, it's one type of the phagocytic cells. Phagocytic cells, the cells who eat others. Why do we call it different words? Just why don't we call it Pac-Mans? They look like Pac-Mans.
neutrophils, eosinophils, basophils, and dendritic cells.
That's so new to me. So I always thought that dendritic cells is part of the brain matter. But if it's phagocytic cell, meaning dendritic cell can eat something alive.
When a pathogen
I'm gonna ask. Okay, I have some chats here. That's doing research. That's also doing something. Two researchers running. I'm sorry. So if then dendritic cell, if dendritic cell is phagocytic on itself, the type, right? The cells that eat pathogens can engulf and eat pathogens alive. If dendritic cell is that type, is type of macrophages, I always thought that dendritic cells, it's a type of our brain matter. So, but if it part of macrophage, not part of the, like, not sibling, the type of macrophage, then our brain matter can eat something alive? I'm really bad at structuring my sentences because I don't have vocabulary, especially pronunciation. So it's hard for me to like output what I'm trying to say. Good question. And there is a mix-up words untangled here. Dendritic cells versus dendritic and dendrites. It's two different two different types of cell. There are two completely different things that share a name because they both looks oh my god. Stop naming shit. Like, okay, I'm sorry. I didn't meant to be mean or something. You guys spent a lifetime on this. But for me, someone new, I just jump into crazy conclusions just because they look alike. Ah. Then the rights are the branching projection of neurons, projections in the brain and nervous system. They receive electrical signals from other neurons. These are not cells themselves, just part of a neuron. Oh. Uh-oh. I have no idea what's our brain made of. We found the big gap. Dendritic cells are immune cells found in skin, mucus, membranes, lymph nodes, and other tissues. They have nothing to do with the brain. So your brain matter is not eating anything alive. The name overlaps. It's just unfortunate. Got it.
Enters the host, and dendritic cells.
Hmm?
When a pathogen enters the host and begins
Pathogen, bacterium, macrophage. First of all, it's way bigger. Awesome. Good job for you. Good for you, macrophage.
to
Phagocytosis.
replicate, it is immediately recognized by one of the macrophages. Membrane protrusions surround the pathogen, absorbing it into the phagosome, which is formed by the fusion of cell membranes. Most pathogens gain access to the host through the respiratory system, gut mucosa, skin lesions, or the urogenital tract. Macrophages are well known to
Whoa. That's so cool for a picture or drawing. So I don't know what it is. Looks amazing.
populate submucosal tissues, and therefore they are usually the first defensive cells to encounter harmful pathogens. Phagocytosis is initiated when certain phagocyte receptors, usually carbohydrate or lipid structures specific to pathogens, interact with the pathogen's surface. A wide range of phagocytic receptors and a variety of signaling cascades can be activated during this process. After being taken into the phagosome through one of several methods of receptor stimulation, the pathogens are eliminated by oxygen-dependent and oxygen-independent attacks. Within the phagosomes,
That's so cool. Oxygen dependent and oxygen independent. So if the pathogen need oxygen to breathe, it basically suffers them. And you're like, if pathogen doesn't need oxygen to breathe, then it's actually put more oxygen. So it's also, that's my guess.
NADPH oxidase produces reactive oxygen species, and phospholipase A2 produces free fatty acids. These molecules are extremely effective in killing the pathogen.
I was wrong.
This is followed by actions of
Look at this guy. Look at him. That's a folded protein, I guess.
myeloperoxidase, or MPO, in addition to various hydrolases and lysosomes, such as azurophilic granules that fuse into the phagosomes, which ultimately degrade pathogen fragments. Next, the proton ATPase pump reduces the overall phagosomal pH, thereby activating either a strong oxygen-dependent sterilization system, mainly through halogenation, or an oxygen-independent sterilization system using
It's chemistry again. Like, I'm so lost, but it's so cool.
lysosomal enzymes. In addition to pathogens, macrophages also remove apoptotic cells in the same manner. Despite their phenotypic plasticity, macrophages have been of immense interest to researchers as being valuable therapeutic targets in various disease settings. Several elegant strategies have been pursued with the aim of functionally manipulating.
Is that the strategies that I just researched? functionally manipulating macrophages so it can be used as different mechanism.
Macrophages. Elegant strategies have been pursued with the aim of functionally manipulating macrophages. Of these, their re-education and
Called it. Yep, I called it. Change the function of macrophages to engulf something else, and you got it. Re-education. They call it re-education.
total depletion can be considered some of the more successful strategies. Macrophage re-education has been used in the field of anti-cancer therapy, in which M2-like pro-tumor TAMs are reprogrammed as M1-like macrophages, endowed with anti-tumor functions. Macrophage depletion is a more extreme approach used to block macrophages, thus directly killing them off. Depleting antibodies such as anti-colony stimulating factor 1 receptor or anti-CSF1R, or molecules.
No, that's completely something else. They like play around and poking with the function of macrophages. Not the way how I thought. And also they trying to eat the tumors cancer. I have a strong belief that vision or whatever. I'm on the side of visual people who think that it's the tumors and cancer. If we try to develop a drugs that's removing the tumors or eating it or stuff like this, we treating it as a symptom. And the real cause is just cells going mad a little bit. They basically lose their concept of behavior, what they should do, and they become barbarian. They become not civilized. So normal cell is behave in environment according with its function. So it's civilized cell. It's knows it lives like somewhere and it's communicate like basically it has its function. But when cancer happen, the cells is just like I'm a amoeba. What I do, I eat and divide and like replicate. And I think we need to fix the brain of cell when it's losing its brain and become cancer, not actually try to clean up symptoms by eating those mad cells. Obviously, we need to clean them, right? But I don't know. It's a deep topic, very sensitive to me. So I need way more education to be able to talk about it.
Exerting a specific toxicity against macrophages, such as bisphosphonates like clodronate and zoledronic acid, as well as trabectedin, and look at some more immune cell types.
Okay, we look at this in the beginning, and I had no idea what it is. Now I think maybe I am. No. Oh, stem cells. Hematopoietic stem cells. Macrophages. That's how they get macrophages. This one going in the blood, right? One of them, and can become this. Okay, I'm going into YouTube black hole. Stop me. Stop me.
He knows a lot about the science stuff, Professor Dave explains. In the previous tutorial, we began looking at the various types of innate immune cells.
Mast cell, macrophages, dendritic cell. I know these two now. I still have no idea what's T cell. T cell is so cool. Everyone talk about T cells. No idea how it functions.
With macrophages covered, let's move on to dendritic cells, abbreviated as DCs.
We got covered macrophages. We learned something today. Macrophage. Now we're going into DC.
Dendritic cells are capable of capturing and presenting antigens to the T cells of the adaptive immune system. The downstream effect of such antigenic presentation
DC precursor. DC, it's a dendritic cell, right? Capturing and presents antigens to T cells. Oh, so it takes that protein from a pathogen. Damn, I speak biology now. So DC takes that antigen from the pathogen and give it to T cell and say like, hey, T cells, remember this is bad. Kill it when you see it next time.
of the adaptive immune system. The downstream effect of such antigenic presentation to T cells can lead to two outcomes: potent activation, known as immunogenicity, or inhibition, known as tolerance of effector immune functions. Activation of the adaptive innate immunity includes induction of CD4 plus T and CD8 plus T lymphocyte activation, as well as B lymphocyte maturation. In contrast, inhibition of adaptive immunity results in peripheral immune tolerance and maintenance of immune homeostasis. Immature DCs are continuously produced from the CD30
Bone marrow. Remember when I said like, look at this, kill it? I was wrong. There is two types. Either adapt or kill it, maybe. On a day 3, that's my understanding. So DC, dendritic cells, which type of macrophages live in bone marrow. But they're premature. Remember, they're premature. So they kind of in a little bit stem cells version. Immature dendrit cell. Hematopoietic stem cells.
34 plus hematopoietic stem cells within
Hematopoietic. I don't know on which day I'm going to remember that word. Hematopoietic stem cells. CD34. Obviously, everyone knows this. I don't know, Dave, how you know this. Like, so cool.
the bone marrow and are found within lymphoid organs, epithelia, connective tissue, and the lymph. Multiple DC subsets have been identified in both mice and humans, and the current classification is quite nebulous, while also rapidly evolving. However, a combination of ontogeny studies and gene expression profiling have provided some insight into the variation within this cell type. The DC family
CDC1, CDCD,
consists
CDDC.
of the following categories: conventional or classical DCs, or CDCs, plasmacytoid cells, or PCs, Langerhans cells, or LCs, and monocyte-derived cells, or MCs. CDCs, also called classical dendritic cells or myeloid dendritic cells, are characterized by the expression of high levels of CD11C and display a characteristic morphology of long dendrite extensions. There are two primary CDC subpopulations in mice, CD11B- and CD11B+. In humans, CDCs express not only CD11B and CD11C, but various other differentiating markers such as CD1A and CD14, to name a couple. Murine PDCs, or those found in mice and related rodents, express low amounts of CD11C and express other markers such as Siglec-H and LY6C. Human PDCs express CD4, CD123, HLA-DR, and blood-derived cell C, located in the oral mucosal action for pathogens such as viruses.
Viral bacterial recognition. Okay, so this is antigens, right? On top. On the bottom, it's a pathogen. Pathogen is antigens sticking out from a pathogens. Am I right about this?
And bacteria. Activation and surrounding environment.
Flagellin. Yeah, flagellin.
For pathogens, such as viruses and bacteria. Activation and maturation of DCs is initiated after capturing appropriate antigens or inflammatory cytokines.
So they mature when they start working. Interesting. Before they kind of like limbo, and when they start eating stuff and capturing, they mature.
Mature DCs then perform their primary task of antigen presentation at their cell surface using MHC molecules. This step is crucial in mounting the acquired or adaptive immune response by interacting with lymphocytes, and this interaction occurs in the lymph nodes. CDCs present antigens and produce the stimulatory mediators TNF, nitric oxide, and IL-12. They also recognize both bacterial and viral antigens. Immature CDCs patrol various tissues to sample foreign antigens. During maturation, CDCs migrate over to draining lymph nodes in order to present pathogen-derived peptides to T cells. PDCs can also act as antigen-presenting cells, but they are much less efficient than CDCs. PDCs predominantly recognize viral antigens, specializing in the production and secretion of type 1 interferons, IL-12, IL-6, TNF-alpha, and pro-inflammatory chemokines. PDCs enter the lymph nodes directly via...
So they're basically signaling for like really bad guys.
via the bloodstream. Alterations in DC function originating from genetic and environmental factors may lead to the development of autoimmune diseases. In autoimmunity, DCs act as a double-edged sword.
I had autoimmune diseases when I actually no, I thought I had the autoimmune, but actually I had a life disease for almost a year.
On one hand, they mount adaptive self-reactive responses, but on the other hand, they promote and maintain immune tolerance. Disruption of the tolerance network that relies on multiple DC subsets can promote autoreactive T cell responses and pathology. Therefore,
T cells again. I need to know what's T cells. Okay, guys, T cells for tomorrow. Maybe we'll read some research. We'll see. For day 4. Day 4 is gonna be interesting, as always.